Okay, now that we've talked about several modes of inheritance, I wanted to take a slight side trip, a very important one for a couple of talks here. I want to talk about multiple alleles. And what we're trying to do here is make you realize that there are lots of variations that can occur, lots of mutations that result in variation at the phenotype, and that these can be out there in various forms. Because, you know, most of the way we teach genetics, at least at this level, transmission genetics is big A, little a, and you get to thinking there's curly and smooth, or there's red and white, and that's pretty much it. It turns out in the real world, we've got a very different system. We have this idea that we have multiple alleles. So I'm going to use a little example here. We're going to start off with some silkworms, these little guys that kind of look like this. Very important, obviously, for making silk, which is still very important today. There's silk farms throughout the world where people work and use the animals onto their native vegetation and then collect the silk balls. And those silk balls then can be pulled apart and made into thread and can make very beautiful garments. So my favorite shirts are silk. They make things like travel bags. If you're travel around the world, or you just go, well, you didn't have to go around the world, but if you travel places, there's these little small bags made out of silk that you can throw out either on your bed or in your sleeping bag. I use them all the time for traveling. Really comfortable. And silk is a wonderful material because, when it's cold, it provides warmth. When it's hot, it can be used. It's really useful for shedding heat. So they're sort of nice little items to carry around. You might want to think about it if you're going to be out traveling the world, particularly as students when we often stay in hostels and things. These can provide some cover. Also protect you from things like bed bugs. I've used them as biologists. We tend to sometimes not stay in the best places, and I've slept in my little bag, silk bag, on top of the bed because I felt that was safer than in the bed. But anyway, they're part of there. And just for as a history lesson, silk was incredibly important. It was one of the major fabrics besides cotton and wool that we had a century ago. Certainly the first part of the 1900s, it was one of the major things, and it's been a common material for clothing for thousands of years. Very strong. I mean, it's stronger than steel per its size. And then when we got into, of course, the Second World War, there have been nylon and other things developed, but silk was still really important, and it was used for parachutes and used for all sorts of things. And there became a shortage of it, and actually, women who had hose that no longer had nylon or silk hose to wear, and so they started to do all sorts of things like draw lines down their legs to look like the hose, because silk hose at the time were sewn and hooked together in the back. And they didn't have any coloration, so they would use all sorts of things, tea bags sometimes to make their skin and things. Why am I telling you that? Well, just from the lack of silk and not having these things, is sort of developed this secondary industry of these tanning materials. So these spray tans and all the chemicals that we have today were actually originally developed because people wanted to have this tanned look during the before and after the war, and didn't have the stuff to cover it up. So the chemical companies and companies responded and came up with these items. Now, it's a history lesson, but let's think about our silkworms themselves. So, if you're trying to use silkworms, you're obviously doing breeding, right? They're domesticated in some sense, at least brought up in a farming level type of thing. And when individuals do that, as we saw back here, you can see that there's sort of these completely light white forms that look like this. You get striped forms, and then you get this other common form called mottled or form, and it's really the mottled is got this sort of blotchy sort of appearance to it. Okay, we're going to call that spotted. Now, if we think about the crosses, it turned out when they were trying to do these early on and thinking about the genetics of these, they would do simple crosses. And they cross a striped with a plain. You get a striped, you cross those F1s and you get 3-to-1 striped plain. Very Mendelian, right? I mean, one of the forms disappeared, in this case, the plain. It reappears in the F2 and it's part of this 3-to-1 ratio, so it's classic Mendelian. But you also had the spotted form, and when you cross that with a stripe, you got striped. And then when you cross those stripes, you got another 3-to-1 with this spotted showing back up. Okay, so that looks Mendelian as well. And even when you took a spotted and a plain, you got spotted, and you ended up with another 3-to-1. So, the question is, what's going on here? We might ask it in a genetic problem. Tell me, how many loci do I have involved? How many alleles? What are the relationships of the alleles? In other words, what's the modes of inheritance that are going on here? Well, the first thing we would do in any problem is pull away the kind of the given information. And what's given here is that we see three phenotypes, right? We see this striped, spotted, and this plain here. Okay, and we don't really see any, we don't have any numbers at this point other than 3 to 1s. But by looking at these 3 to 1s, what have we seen from those? Well, they're simple Mendelian, right? So it looks like we've got just a series of simple Mendelian systems going on. Well, what's really happening here is pretty simple. Okay, what's really happening here is we have one locus, so we don't have anything different than big A, little a, and all that. But now we have three alleles. Okay, three different alleles, three different mutations that occur sort of at the same time. So what this way this is working, because of in this case we have this sort of dominance is what's known as a dominant series. So we have three alleles. The three alleles are striped, spotted, and plain. So the striped allele causes you to be striped, spotted causes you to be spotted, plain is plain. But they're in a complete dominance relationship, okay? Striped is dominant over both of these. So if you get a striped allele, you end up being striped, just like here, right? If you get a spotted allele, it depends on whether it's with a plain or a striped. If it's with a stripe, then the stripe dominates. If it's with plain, then actually it dominates. It does something like this. So it's known as a dominant series. And if you go back and try to work this problem, I think you'll see if these are classic parentals, so they're true breeding, right? These will all breed true, right? This spotted right here would be S prime, and then little s. We get a heterozygous individual here that's S prime, S, little s. But the spotted dominates over plain, so you don't see plain here. Cross those hets and we get a nice 3 to 1 outcome, and that's the same for all of these. You can fill them in with these alleles, starting with homozygotes, classic parents over here, true breeders, making hets, crossing those hets, and giving these 3 to 1s. Okay. Now, if we think about it, and we think about what a gene looks like, a real gene, a piece of DNA, we've got all this code, ATGC, ATGC. This is just a segment of DNA from somewhere that's part of a gene. These little letters represent the amino acids that would be put into the protein, this code. But any place we change in this gene, and this is a fairly small one. What have we got? Only about 3,000 bases illustrated here. Any place we change that T right there to a G, we've got an allele. We change that T to an A, we've got another allele. We change those, we've got an infinite number almost of changes that can be made here. And each time we make a change, they might well represent a change in the phenotype, right? So we have one locus. Because that locus represents a gene that may be several thousands of bases long, and any one of those nucleotides could change, we have just a massive number of alleles possible. And that goes back to our early discussions when we talked about DNA and what a genetic, any kind of good genome needed to be. It needs to be able to store massive amounts of information. Well, there's an example of ability to store massive amounts of information and to be variable about it. Now, fortunately, we do see a lot of changes when we'll see them in lab when you compare pieces of DNA from different organisms. We see a lot of differences, but most of those differences don't end up being phenotypic changes like this going up over here. They may be molecular level alleles because they're mutations of the DNA. They don't necessarily correspond or produce a what's known as a phenotypic allele, a phenotypic variant. Allelic form. And that's because we have a lot of silent ones. We have complex systems where the product from this gene has to interact with another, and it may not matter, etc. Okay, but we do see multiple alleles in populations. This is a little example from giraffes, a real world example. We have this sort of big A, we just call it little a, big B, little b, and S alleles. And whatever that's all at one locus now with 6 alleles, and whatever combination of those you get gives you a different coloration or striping pattern in giraffes. Okay, so 2 little a alleles kind of gives you this lighter colored one with big stripes. If you get down and have 2 big alleles, black alleles, you end up with the black classic coloration that you see over here. And if you get some A allele, because A kind of is putting in orange, you get the big spots, or the big flanges, but you get sort of the orange part of it. And it's all these different forms. And so all of these genotypes are possible among giraffes based on the fact that there are these 6 alleles present. So you could be big A, you can be big A, you can be AS, you can be BB, you can be SS, you can be any of these forms, all at one locus. So if this giraffe up here, AS, breeds with this giraffe down here, BA, you can see that you got multiple types of progeny, but it's all genetics, simple, basic Mendelian genetics that goes on. And of course, now we actually know now in a very recent research studies that there are actually 4 species of giraffes. We thought there were 4 subspecies, but they've actually now broken them into from one big species into 4. A lot of it based on this pattern of coloration as well as their locations. Just something to look up. Now, other real world examples, your fruit flies that you've been working with. Well, the wild type wing shape, this normal wing that a fly can fly with it, is a dominant, and it dominates over lots of different types of variants. These aren't all the variants that you could have, but you've got vestigial, strand, nicked, notched, and all these. When these come together, you get various forms that are heterozygotes or intermediates in some way. So if you get something like a nicked and a notch together, you'll get one form. If you get a strand in this, but if you have a wild type, it will always dominate over these others, and you'll end up with a normal shaped wing, like that. Clover, good example. Wild clover has 7 alleles, and depending on which combination you get, either as a homozygote, maybe of the little v alleles, you're just pure green, all the way up to all these massive colorations. So next time you're out looking at clover, kind of fun, look at the little pattern of this sort of chevrons patterns that are in the middle, and you can actually figure out from that what the genotype of that particular plant is, okay, by just simply looking at those patterns and trying to think about the 7 alleles. Other places we see it, plants, it's one of the common things in plants. A lot of plants you don't want to have selfing, right? You don't want the plant to be able just to, if the plant's capable of producing both ova and pollen, you don't want the pollen just falling down and fertilizing the plant. Some plants, of course, do that. They do self-fertilization, but others have ways to kind of prevent that so that they maximize the genetic variation. This is known as gametotrophic self-incompatibility. So the self-incompatibility means that if you produce this pollen, it's not going to self-fertilize in the stamen that you have, right? Tobacco is a good example. There are no heterozygotes, homozygotes in tobacco. So there's no S1, 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 S1 alleles for there, okay? And in fact, if we look at the pattern, if you try to cross anything that has the same alleles of these 5 alleles that are quite common, there are 5 alleles that are involved in the system for most tobacco. If you try to put, say, a pollen it doesn't matter where the pollen comes from, the same plant or another that's got S1 and the genotype of the plant itself has got an S1 in it, then it's not going to form a full tube. You're not going to get a pollen tube and therefore not going to get fertilization. Okay? If you have at least one difference, right here we have the pollen coming in as S1 or S2. Remember, these are gametes, right? So they're going to come in as haploid. Well, the S2 won't work because this plant is an S2 allele as part of its genotype, but the S1 will because the other one is an S3, so that one will actually pollinate it. Or if you got complete differences like these two, either one of these pollens could pollinate it, and that's exactly what's happening. You see, it's basically preventing itself from self-pollination, right? So multiple alleles. The basic idea is that systems have a lot of alleles, right? And we have that because the genotypes the genes can just be modified greatly. And some of these actually end up having effects. One that's in humans and one that is important for working a lot of problems would be the ABO blood group, right? And all of us are almost all familiar with that. You either have type A blood, type B blood, type AB blood, or type O blood, okay? But it turns out it's a fairly complicated system because the A allele is dominant over O, B is dominant over O, but A and B are co-dominant to each other, okay? So we have two modes of inheritance going on at the same time. For one allele combination, it's dominant. For the other allelic combination, it's actually codominant. And it's true codominance. Both of these will show antigen-antibody reactions when they show up. Okay, so if we go out and we genotype somebody, they may be AA or AO, which will give us A as the phenotype, A-type blood. BB or BO will both give B-type blood. If we see that an individual in their phenotype is AB, we know that their genotype is AB as well. And O, we know that they're homozygous O because it's the most recessive. That's why O is considered the universal donor, right? It can provide blood to all these types that we have, right? So it's actually, you know, we talked about the MN blood group at one point as an example of codominance, where you have M gives you M blood, or MN gives you MN blood type. Here we've got a slightly more complicated system, but it works out. I mean, don't worry about the genetics of it. It's fairly straightforward. Here's two hets, AB times AB, right? They would expect their progeny to be just like a Mendelian cross. About 1/4 of them are going to be type A blood. About half of them are going to be type AB blood. That's their phenotype, and about 1/4 are going to be type B blood. That's going to be their blood type forms. Okay. And then, of course, if you involve all three alleles, then you get a slightly different combination, because now you're going to be putting A with A, and sometimes O with A, sometimes A with B, or O with B. And when you do that, genotypically, you get the outcomes you expect: one AA, one AB, one AO, and one BO. But now, because of dominance, right, this guy is going to be type A, that one's going to be type B, that's going to be type A, and this one's going to be type AB. So the phenotypic segregation ratio, this is segregation because it's only one locus, the ratio we would see would be a 2 to 1 to 1, right? Or 1 to 2 to 1, however you want to put, what order you want to put it in. So once again, we've got this, just because you've added an allele, no difference. Okay. All right, well, that pretty much introduces multiple alleles. And like I do with every lecture in transmission so far, I try to introduce the idea with one or two of these smaller lectures. And then we go to the final part of this, we try to work problems, right? So the next part of this, part 2, we're going to take this multiple allele concept and do some basic problems that you might see either in textbooks or web, or something you might see on an exam.